Good afternoon, ladies and gentlemen. My topic is development and application of single-cell omics sequencing technology based on non-read platforms. Single-cell omics technologies consist of many modalities, each focused on a part of the central dogma of DNA and RNA. We can use various tools for multiple purposes, including quantification of genome, exome, and epigenomic, transcriptomic, and epitranscriptomic from information from DNA and RNA. Joining these single-cell omics views with computer gives us huge help in basic and medical researches. Precious analysis of molecular and functional heterogeneity can make analysis of cells and tissues to understanding disease better. Here are some key technologies that have allowed jumps in experimental scale. There are many disadvantages of single-cell RNA-seq based on NGS compared to third-generation sequencing, including only obtain gene expression levels data, false positive spelling sites, and lack of transcript diversity. The concept of transcription encompasses not only the abundance of gene expression, but also the specific structure of transcript sequences. Alternative splicing enables a single gene to generate multiple transcript variants, which can be translated into diverse proteins, thereby enhance functional diversity and enabling a gene to participate in various biological processes. Alternative splicing plays a critical role in development, regulation, and disease pathogenesis, such as the selective usage of transcription start sites in neurons, the development of gene isophones in signaling and value sensing. The advent of third-generation single molecule sequencing has opened up opportunities for analyzing known fragments, a technology that was notably recognized as the method of the year 2022 by Nature Methods. The rapid development of NGS-based single-cell RNA-seq allows for detecting and quantifying gene expression in a high-throughput manner, providing a powerful tool for understanding cellular function in various biological processes. However, the NGS-based single-cell RNA-seq only quantifies gene expression and cannot reveal the exact transcript structures of each gene due to the limited readiness. The long-read length of TGS technologies, including LLPO and PacBio, enable direct reading of intact cDNA molecules. Both LLPO and PacBio have been used in conjunction with single-cell RNA-seq, but their performance in analysis has not been systematically evaluated. We collect embryo, lung, and brain samples from single-cell transcriptome amplification yield as 500 or 5,000 cells. cDNAs were divided into 3 groups subjected to NGS, LLPO, and PacBio sequencing platforms. The read quality of PacBio was comparable to that of NGS, and LLPO reads were obviously below. Only about half the LLPO reads can obtain full-length transcripts, and the majority of the PacBio reads can be split and derived full-length transcripts. For cell barcode detection, PacBio outperformed NGS in perfect matching to the whitelist. In contrast, less than 16% full-length transcripts captured by LLPO contains cell barcodes perfectly matched to the whitelist. Unsupervised casting and cell type identification was carried out using single-cell gene count metrics from 3 sequencing methods and isoform count matrix from 2 TGS methods. We identified 5 cell types in all the datasets, except that muscle cells were not identified in the NGS 5 hectare sample. The cells identified in the TGS datasets were casted into mechanical cells in the sample. This suggested the TGS platforms performs better in cell type identification when the sequenced cell number is limited. With the increased sample size, the NGS captured the cell types more effectively. By gene count matrix, the same cells were more consistently assigned of the cell type identities between NGS and PacBio. The same cells were almost identical to the same cell type in both TGS platforms through isoform level casting. Although the TGS showed no gene detection ability in single cells of the sample, they didn't cause effect on cell clustering and cell type identification at both gene expression and isoform expression level. The expression level of marked genes for each cell type were nearly identical between the 2 TGS technologies. There was difference at the isoform expression level. For example, PacBio, but not NelPo, detect more TNT2 transcripts in muscle cells. The PacBio data was obtained with more cell type differential experimental genes than the NelPo data. More cell type DEGs and DEIs were obtained when more cells were captured in a dataset. NGS yielded the highest number of DEGs, followed by PacBio, with NelPo skipping the lowest cell type DEGs and DEIs. To evaluate the ability of detecting isoforms by each TGS method, we sort genes by the number of detected isoform types. Some genes were detected with more than 100 isoform types, and over 1,000 genes were detected with at least 10 isoform types. However, the reliability of isoform needs further discussion. A higher number of lower transcripts were observed by PacBio, and there was greater concordance in the lower transcripts identified between different methods with the same sample. We then evaluated the activity of lower isoforms captured by each TGS methods. A higher rate of PacBio-specific than NelPo-specific lower transcripts were validated, suggesting higher accuracy of PacBio in identifying lower transcripts. These results support the utility of TGS technologies in unraveling the complexity of transcription landscape, highlighting the better performance of PacBio in lower transcript discovery. We also studied antigen-specific gene expression. The full-length transcripts directly read by TGS afford us opportunity to do such investigation, as we cause 2 different strengths of mice. For sampling, we could figure out the antigen-specific rates according to the single nucleotide parameters between the 2 strains. Consequently, more reads in PacBio data can confidently assign to any origin than those in the NelPo data for each cell type. For instance, SFTP1, a classical marker gene of alveolar epithelial cells, contains 2 isoform types. TGS detected no difference in expression from both ends for whole gene, but the anions express more SFTP1 transcripts, and the DBA2C anion express more SFTP2 transcripts. This indicates that the SNPs on the gene may affect the spelling of the gene. These results suggest that there are biased gene transcription and spelling between the two areas of mouse genome, which can be confidently captured by PacBio. And we comparison the cost of two technologies. We find that more cells in a library can decrease the cost per each cell. Based on our evolution, both NanoPro and PacBio achieved comparable results with NGS on cell type analysis, with only a twofold increase in cost. Moreover, they offered supplementary insight into isophones and unique expressions. The reduction in TGS-based single-cell RNA-seq price would enable more researchers to afford studies and facilitate broader scientific expression and discovery. Oncogenes often amplify on extracellular chromosome DNA in cancer, but we don't fully grasp ecDNA structure or how it influences gene control. ecDNA impacts oncogene and links ecDNA effects with today's cancer's genetics and epigenetics knowledge. In design, we use magnetic beads to separate DNA and RNA molecules into each single cell to obtain the DNA and RNA for downstream amplification and sequencing. Then SMART-Seq2 was applied to amplify the RNA in cells, and the adjusted smooth-seq products and library construction for TGS were performed with the genomic DNA. And we established a bioinformatics pipeline named ecDNA Finder and joined exogenomic plasmid and verified the count of constituency. To validate ecDNA candidates are real ecDNA, we label the oncogene MYC located in ecDNA with fish probe tiny on 200 kilobase pair sequence in the head, tail, and the middle parts of the ecDNA. The results showed that the single-cell GTP-seq workflow combines with our pipeline enabled the identification of endogenous ecDNAs. We also found numerous ecDNAs within the region overlapping the MYC gene in 3 cell types. This indicates that there might be some ecDNA hotspots in cancer cells. As expected, the ecDNA genes had a significantly higher expression level than non-ecDNA genes. And our data suggests that the copy number may not be the only factor that determines the transcription level of ecDNA genes. Compared with the in vivo cultured cancer cell types, we apply single-cell GTP-seq to colonial tumor. The single-cell GTP-seq data revealed expected CNV events in hepatocells and non-cancer cells detected with normal diploid chromosomes. We selected some genes such as MYC, which was only present on ecDNAs in hepatocells for experimental verification using fish. The copy number of the MYC gene was higher in part of hepatocells compared with both TECs and TAMs. ecDNA plays an important role in tumor genesis, development, diagnosis, and treatment. Here we outlined the research trajectory of ecDNA, discussing its role as a genetic and prognostic biomarker, and illocate its electronic mechanisms in cancer. We highlight its possible use in cancer diagnosis and treatment, and looking forward for lower tumor diagnosis strategies using blood ecDNA with PacBio in the future. family, we thank my professor Lo Guanzheng and Xiao Chuanle, and my supervisor Xiao Yifan. Thank you for your time.